today I'm going to tell you about the rules of phosphorylation and protein folding in regulating RNP granule condensates during oogenesis. My lab has had a long-standing interest in understanding how the regulation of RNP granules may affect oocyte quality. On this slide, I'm showing how oocyte quality and quantity change over time. A woman's age is shown on the x-axis. The dark black line indicates the dramatic decrease in the number of follicles that occurs after a woman is born. The red line shows the sharp increase in the proportion of poor quality oocytes that occurs after around the third decade. The causes of poor quality oocytes include chromosomal abnormalities and organelle dysfunction. And today I want to introduce the hypothesis that the dysregulation of membraneless organelles also contributes to poor quality oocytes. Now, to consider this hypothesis, we first need a working definition of membraneless organelles, also known as biocondensates. In contrast to membrane-bound organelles, which are shown on the left, where the membrane generates a compartment boundary to organize cellular contents, membraneless organelles, or biocondensates, shown on the right, can organize cellular contents via a process called phase separation. So these compartments are formed by phase separation, which also generates spatiotemporal control over diffusion-limited biochemical reactions. Now, over the past decade, many examples of membraneless organelles have been identified and characterized. These include nucleoli, stress granules, and germ granules, among many others. This process of phase separation is a very powerful mechanism to generate cellular compartments. However, it is vulnerable to changes in chemical and physical parameters. For example, changes in the concentration of RNA and protein, changes in pH or cellular energy levels, can all result in condensate components undergoing phase transition among differing biophysical states. To illustrate this point, here I'm showing you in vitro synthesized fluorescently labeled FUS protein, which is shown in green in a diffuse or dispersed state. In the cartoon, the protein is indicated by the red lines. In this state, the protein has a high level of dynamics. Under certain conditions, the protein can undergo phase separation or condensation to form liquid-like granules. This is a reversible process. Further condensation can occur to generate gel-like granules, which have even reduced dynamics relative to the liquid-like granules, and this is still a reversible process. A solid-like state can also occur, which is not reversible, and this state is sometimes associated with pathological conditions. For example, in ALS, aberrant phase transitions of the FUS protein occur, changing it from a liquid droplet to a solid aggregate, and this contributes to the loss of motor neurons. In the germline, fewer examples have been identified to date. However, nuclear condensates of the FMR poly G protein occur in FXPOI patients, as shown on the right. So in Fragile X-associated primary ovarian insufficiency, there are increased rates of infertility in women that are not completely understood. These examples, as well as many others, clearly indicate the regulation of phase transitions is critical for cellular function. Now, what kind of biochemical reactions might be regulated by phase transitions in the oocyte? A recent paper from the Afruzi lab beautifully demonstrates that biophysical states of condensate components can modulate RNA localization and translation. This study was done using the Drosophila oocyte model, which is shown here at the top. On the left, you can see RNP granules or biocondensates, and these are solid-like granules composed of the Oscar RNA and RNA-associated proteins. When these investigators perturbed the solid state, changing it to a liquid state, they observed impaired RNA localization and translation and defective embryonic development. This is a really beautiful study and is one of the first to clearly indicate that maintenance of the solid state or phase is equally as important as maintaining the liquid phase depending on the cellular context. In addition, the study clearly shows that the function of RNA and RNA binding proteins is dependent on the biophysical state. In mammalian oogenesis, we don't yet have a mechanistic understanding of phase transitions. However, transient RNP granules have been identified. On the left, I'm showing oocytes from primordial follicles of neonate mouse ovaries. So in green is the conserved RNA binding protein trailer hitch, and in red the human homolog RAP55. These proteins co-localize in the Balbiani body. which is one of these RNP granules that is transient and actually disperses in later oocytes. A second example are the subcortical aggregates observed in oocytes from 6 to 14-week-old mice. So here in the top, we see GV stage oocytes, where the RNA binding proteins DDX6 and MSY2 co-localize to these subcortical aggregates shown by the arrowheads. These are transient structures. In meiosis 2, you see that these proteins have decondensed. In addition to the RNA binding proteins, untranslated mRNAs also localize to these subcortical aggregates. In human oocytes, Peewee-like proteins have recently been shown to transiently condense into granules as well. Here I'm showing Peewee-like 1 protein. It condenses to a single large granule in premeiotic oocytes. The protein decondenses during pachytenomyosis and then recondenses into a granule in oocytes of primordial follicles. Now, this study was done using second trimester fetal gonads, so we don't yet know if these granules change over time with age, or if they might have any function in regulating oocyte quality. So I hope by now I've piqued your curiosity about RNP granules and the regulation of phase transitions, and convinced you that investigating them during oogenesis is worthwhile. We know these granules and their components are highly regulated throughout Drosophila mouse and human oogenesis, and the function of RNA and RNA binding proteins can change as the biophysical state changes. Ultimately, we'd like to understand to what extent does regulation of condensation promote oocyte quality? Does it impact RNA stability and translation and ultimately oocyte quality very broadly? And as a step towards understanding and addressing this question, my lab has been using the genetic model system C. elegans to characterize how phase transitions of RNA binding proteins are regulated during oogenesis. So C. elegans is a nematode genetic model system. It's especially effective to study the regulation of phase transitions for many of the typical reasons genetic model systems are powerful. It's a small size, has large numbers of offspring, and a rapid life cycle. In addition, C. elegans exists as self-fertilizing hermaphrodites and males. It's also optically clear, which is shown in this light micrograph of a living hermaphrodite on the right, and you can see I've bracketed the 2 rows of oocytes in the anterior and posterior gonad. There are also well-developed genetic tools, including RNA interference to deplete gene expression and CRISPR, which is useful to generate fluorescently labeled reporter strains for live imaging. I need to introduce you to the anatomy of the C. elegans germline. So at the top of the cartoon, I'm showing you the 2 U-shaped gonad arms that meet in the common uterus in the center of the worm, where the embryos are found. If we zoom in on one of those gonad arms, you can see at the top left, in blue, it's labeled mitosis. This is the distal end of that gonad arm and a self-renewing population of germ cell precursors. Those cells fairly rapidly transition into meiosis, and then are found in pachytene until they round the bend of the gonad arm, where they progress into diploatine and diakinesis of prophase 1. At this point, these prophase 1 oocytes are ready for meiotic maturation and ovulation, and they will move into where the sperm is stored to be fertilized. Now, in a young hermaphrodite, this process is very active, and meiotic maturation occurs every 23 minutes. In these diakinesis oocytes, there are 2 populations of RNA binding proteins. The first population has a default state of being somewhat condensed and forms small germ granules. Proteins that localize to these germ granules include GLH1, homolog of VASA, DDX4, and 2 C. elegans specific proteins, PIGL1 and MEG3. The second group of proteins has a default state of being relatively dispersed or diffuse, and proteins in this category include CGH1, homolog of DDX6 RCK, CAR1, homolog of LSM14, RAP55 trailer hitch, and MEX3, homolog of MEX3 A to D. So in a young hermaphrodite, in these diakinesis oocytes, there are these 2 different states of RNA binding proteins. So how does meiosis change during aging, and do these phase transitions change? And the answer is that many changes do occur, including an extended meiotic arrest that occurs in middle-aged hermaphrodites. And this is one of the reasons that C. elegans is an especially powerful system to study oogenesis. So in contrast to the young hermaphrodite on the left, where sperm are present and meiosis is active, during middle age, the sperm get depleted. They get all used up, and therefore the oocytes no longer have the signal to undergo meiotic maturation and ovulation, and so they stack up in the gonad arm. So in this case, they are undergoing an extended prophase 1 arrest that can last for several days, which is a significant percentage of the worm's lifespan. Similar arrest can occur in FOG2 mutants, which don't make any sperm. So in those female worms, there's an extended prophase 1 arrest during the entire lifespan. This arrest is reversible if the worm is mated by a male and sperm is replenished, then meiosis resumes. So the reason we're very interested in these arrested oocytes is because of the dramatic phase transitions that occur in arrested oocytes. Now, before I tell you about those dramatic phase transitions, I just want to point out the evolutionary conservation of this prophase 1 arrest. So in contrast to the variation we see with secondary arrest during meiosis, which can occur at many points during the cell cycle, primary arrest occurs in all species at prophase 1, and it's often released by hormones, and in C. elegans that release occurs by a signal from the sperm. So this conservation really underlies the utility of using a really simple model system to understand oogenesis. Okay, so what kind of changes occur in these arrested oocytes? What we observe is large-scale condensation of RNA-binding proteins into large RNP granules during extended meiotic arrest. Both the condensed or germ granule proteins as well as the diffuse RNA-binding proteins undergo this large-scale condensation. So these RNP granules include germ granule proteins like PIGL1 and MEG3, those diffuse or dispersed proteins like CGH1, CAR1, and MEX3, and many untranslated mRNAs, including POS1, which I'll show you here. So this confocal micrograph shows you the difference in size of these granules. In the top row are small germ granules from diakinesis oocytes, where we see POS1 in red and PIGL1 protein in green co-localizing. And then in contrast, we see very large granules that can be up to 20 times larger than germ granules. So these are the large the large RNP granules in arrested oocytes. So based on their composition, we hypothesize these large RNP granules may maintain the quality of oocytes during the extended meiotic arrest. Furthermore, based on their components, we speculate the regulation of RNA metabolism may be altered in these arrested oocytes, requiring this phase transition. Not shown on this slide is the fact that this condensation is reversible. If meiosis resumes, because the worm is mated, both the RNA and RNA binding proteins decondense rapidly. So this is a very dynamic process. So to better understand these RNP granules, we've recently undertaken several functional assays to characterize different RNA binding proteins within them. So here I'm showing you FRAP analyses to study protein dynamics. The first two proteins I'll mention are PIGL1 and CGH1, which you can see in the top line of the graph, those lines merge, have fairly rapid recovery after photobleaching. In contrast, MEX3, shown in green, has slightly slower recovery rate, and MEG3, shown in red, has very slow, if any, recovery at all. So these four RNA binding proteins have distinct dynamics within these RNP granules, suggesting they may occupy distinct phases. A second assay we've used to study these RNA binding proteins is sensitivity to elevated temperature, because sensitivity to high temperature is often associated with liquid-like phases. So here we're looking at some live imaging where we're using GFP-tagged strains of PIGL1, CGH1, and MEX3. And in the top row, we can see in those arrested oocytes, those very large granules indicated by the white arrows, as we would expect. After an upshift to 34 degrees, we can see with the yellow arrow for PIGL1 that there is a dramatic decrease in condensation. Although we can still see faint outlines of those large granules, the majority of the protein is no longer condensed. For CGH1, we see only modest decondensation. The majority of the protein is still in those granules, as we can see with the white arrow. And for MEX3, we actually detect further condensation as determined by our quantitative analysis. So only PIGL1 appears to be very sensitive to elevated temperature. We've also done other assays, including hexane diol and other stress assays that I don't have time to show you today. But altogether, these different functional assays lead us to a continuum model of distinct phases within RNP granules. So in this kind of blow-up cartoon of a single granule within an arrested oocyte, MEG3 is in red, MEX3 in green, CGH1 in dark blue, and PIGL in light blue. And we are speculating that these distinct functional phases within RNP granules may correspond to cytological substructure that has been observed for many years, as well as potentially different functions. Moreover, we show at the bottom of this model that while these 4 different proteins occupy distinct locations along this continuum of liquid-like to more gel-like phases, with PIGL1 being the most liquid-like and MEG3 the least liquid-like, that there's an inverse correlation with their stability during stress. So we're very interested in how this condensation is regulated. We can see that it's very dynamic. We can see that these RNA binding proteins have some distinct functions. Overall, how is this process being regulated? And based on these data, we wondered if the more gel-like proteins, MEG3 and MEX3, might play a scaffolding role to promote condensation of the liquid phases, PIGL or CGH1. However, we found no evidence to suggest this was the case. I'm going to use my remaining time to tell you about 2 regulators we have identified, and I should say that RNA may also have an important role in molecular scaffolding or nucleating, and that's something else that we are planning to follow up. But as I mentioned, I want to tell you about 2 regulators today that we have identified, and the first is a post-translational modification. So we got our first hint that post-translational modifications might be important in regulating condensation during oogenesis from many in vitro studies. So phosphorylation, ubiquitination, methylation, and many other post-translational modifications have been identified in a series of beautiful in vitro and cell culture studies. So we wonder to what extent this type of regulation might actually carry over to an in vivo context such as oogenesis. Now, how could post-translational modifications actually regulate these phases or properties of condensates? So this cartoon suggests that these modifications can enhance or disrupt the weak multivalent interactions between RNA binding proteins. And in this way, the modification could either promote condensation from a dispersed RBP to a liquid-like condensate, or it could actually inhibit that process. So phosphorylation, for example, has been shown to both promote and inhibit condensation into a liquid-like granule. The first regulator that we have identified during oogenesis is ERK, the extracellular regulated kinase. This is very strongly conserved kinase that has important functions in many cell types. And what we find is that activated ERK is inversely correlated with MEX3 condensation. So in diakinesis oocytes, when MEX3 is dispersed, as shown in panel A, we see that activated ERK is detected, as shown in panel B. We see this in the nuclei as well as in the cytoplasm of these 2 oocytes. In contrast, in arrested oocytes, where we know that MEX3 condenses into granules, in panel C, in panel D, we no longer see activated ERK. So this inverse correlation suggested the hypothesis that activated ERK inhibits the condensation of RNA binding proteins into RNP granules. So to test this hypothesis, we used RNA interference to deplete ERK, and we're doing live imaging here where we're using GFP tagged reporter strains. So here I'm showing you MEX3 and CAR1. In the control images of these diakinesis oocytes, we just see dispersed protein as we expect. And after ERK depletion, we do see condensates. So for MEX3 in the top row, you see the white arrow indicating some of the moderate-sized granules that we detect. And for CAR1, we see smaller spherical condensates indicated by the white arrow in the bottom right panel. And also, the yellow arrow indicates larger condensates that appear similar to ER-like structure. So we're currently following up on what that might mean. We've also detected condensation of several other RNA binding proteins, albeit to differing extents. So it appears that ERK definitely has a role in these diakinesis oocytes in inhibiting condensation. We're interested to know if the canonical RAS-ERK pathway is also involved in regulating this condensation. So we do have some preliminary data suggesting MEK, the upstream kinase that phosphorylates ERK, is also playing a role in inhibiting the ectopic condensation of granules in diakinesis oocytes. And we're going to be following up and looking at those other upstream players in this pathway. We're very interested to understand the mechanism by which ERK is modulating condensation. One model is that ERK is phosphorylating RBPs directly to block ectopic condensation. Secondly, ERK may act through phosphorylation of the CCT chaperonin, and I'll explain why we're interested in this as a possible intermediate on the next slide. A third possibility is that ERK might phosphorylate substrates that modulate cellular remodeling, in particular the endoplasmic reticulum or microtubules. And I don't have time to show you the data that support this idea, but this is yet another model we're interested to follow up. And then lastly, ERK might act through both CCT and cellular remodeling to eventually block the ectopic condensation of RBPs. So why are we interested in this CCT chaperonin? This is a chaperonin also called Tri-C, and it turns out it's a conserved regulator of phase transitions. It inhibits P-body and stress granule protein condensation. It also inhibits polyglutamine aggregates. And in C. elegans, multiple genetic screens have identified distinct subunits of this CCT chaperonin in promoting the condensation of different RNA binding proteins. So we have been interested to understand to what extent does CCT chaperonin function in oogenesis, and could it be an inhibitor of condensation in diakinesis oocytes? We're also wondering if there could be any subunit specificity or any specificity in regards to the developmental context or RNA binding protein. So why are we interested in these subunits? Well, the CCT chaperonin has 8 asymmetrical subunits arranged in 2 rings that form a cylinder shown here in this cartoon. So this is the protein substrates enter into this cylinder and then become correctly folded. This is an ATP-dependent process. So here you can see those 8 subunits shown in the middle cartoon. The mammalian subunit names are shown on the right. And it turns out the CCT chaperonin is very important in our cells, estimated to fold about 10% of the proteome. Its obligate substrates include actin and tubulin subunits. So to test if CCT could be regulating MEX3 in diakinesis oocytes, we undertook a similar approach by using RNA interference to deplete CCT subunits. Here I'm showing you how MEX3 undergoes condensation into large condensates, shown by the white arrow, after depletion of CCT2. We've tested 6 subunits, and depletion of all of them except CCT4 it seems to result in these ectopic granules, suggesting that CCT1, 2, 5, 7, and 8 all inhibit ectopic MEX3 condensation. We next asked if CCT regulates other RNA binding proteins besides MEX3. So here I'm showing CAR1 again, and after depletion of CCT2, we see ectopic condensates forming. We've examined 4 different subunits, CCT2, 6, 7, and 8, and all 4 appear to have a role in inhibiting ectopic condensation of CAR1. We've also asked if CCT regulates germ granule proteins, those proteins that have a default state of being semi-condensed into small granules. And after we deplete CCT2, we see actually some decondensation of the GLH protein, which is shown here on the far right. We have seen actually a mix of phenotypes. So 4 CCT subunits, 1, 2, 6, and 8, seem to promote GLH1 condensation to a certain extent, but CCT4 and 5 do not seem to have this same effect. Interestingly, we were not able to detect any change in the condensation of PIGL1, another germ granule protein. So taken together, we now have quite a lot of evidence that CCT is a major regulator of condensation in oocytes, and our models that we're currently pursuing for how this might be happening are shown here. First, on the left, it could be that MEX3 and CAR1 or other RNA binding proteins are direct substrates, normally folded by this chaperone, and that correctly folded native protein is going to inhibit any ectopic condensation. And this would go along with the general idea that misfolded proteins tend to aggregate. Alternatively, the microtubular actin cytoskeleton might be mediating the regulation of RNA binding protein phase transitions. So we know that tubulin and actin in most eukaryotic cells are substrates of CCT, and perhaps those cytoskeletal elements need to be folded properly in order for phase transitions to be properly regulated. So we're currently pursuing both of these models. And to wrap up today, I hope I've convinced you that phase transitions are dynamic and maybe functionally important during oogenesis. Our results indicate that ERK and CCT are important regulators to block the ectopic RNP granules in diakinesis, so to block condensation when it should not be happening. And interestingly, several CCT subunits have conserved sequences of ERK docking domains and phosphoacceptor sites, so it may be that ERK and CCT are acting in the same pathway. We further propose that in arrested or old-aged oocytes, the situation may be different, right, with this extended meiotic arrest, and these condensate components, the RNAs and RNA binding proteins, may have a requirement for altered metabolism, requiring that large-scale condensation into granules that seem to have both a liquid-like and gel-like phase. And overall, we think that these two different developmental contexts have different requirements for proper regulation of RNA metabolism, RNA stability, and translation, which may ultimately then modulate oocyte quality. I'd like to acknowledge all of the contributions of the students who've done this work. Mohammed Alazwad, a talented PhD student in my lab, Mingzi Gao, talented master's student, and a great crew of undergraduates, Corinne, Cora, Nicole, Nick, and Christia. I'd like to acknowledge our collaborators, Swathi Arora and Deba Das at MD Anderson for their help and support, the CGC for strains, our former students for all of their wonderful contributions, and our funding sources. Thank you for your attention.